Okay, in this video I'm going to show how to make a transgenic mouse. And this is something that we commonly use in research to replicate a human disease and so we can figure out how to develop a cure for that disease. So what you need to do initially is have to have recognize a region of homology. So as you can see in these two strands of DNA, they have the exact same sequence right here in the region of homology, which is GCCCCGAT, and that's going to be the same in both strands. And you can see those nucleotides on either side don't match, so these two are similar. So this is going to happen during meiosis. Crossing over can occur, where you'll have two different strands, and they have a homologous region where the nucleotide sequence is the same, and they can actually flip over like that. So you can see on this strand it's all A and it switches over at this point, causing it to be AB and this one be BA. So knowing that, researchers have developed a way to manipulate this so we can insert a select gene into a mouse model. So initially what you need to do with this is have an engineered vector. So you want to have the two homologous regions which will integrate into the host genome. So these two homologous regions will be the same as the host, and on one side you want to make sure you have the desired gene, so whatever you're trying to insert, whether it's novel like glow in the dark or a different color pigment or a disease like cystic fibrosis, you can insert that. And also you want to have an ampicillin resistant gene. I'll get into that later, as well as a TK gene that's outside of the regions of homology. So initially what you want to do for this procedure is you want to take a brown mouse stem cells, and you have to make sure it's brown because that's going to be a tracer later on, and that's pretty important. So you take stem cells from it and you infect them with the vector, and you put these on a agar plate that has ampicillin. So there's going to be three options three different outcomes that can occur from this. Either you'll have a random insertion where it didn't really follow where it integrated at a wrong part of region of homology, or it just kind of randomly inserted into the genome, and we're going to be able to kill off any cells that accidentally do this by treating the culture with glycovivir. And what this does is this will react with the protein created by TK, causing the cell to die. Another option is there's no insertion, so the cell will die being put on an ampicillin plate because it won't have the ampicillin resistance gene inside of it. And then there's a correct insertion, which is what we want, where it survived being on an ampicillin plate because it has an ampicillin resistant gene. It's recombined correctly, so the TK gene is not inserted into the genome, so glycovivir will not affect the host. So now that we have those cells on the plate, we want to take whatever remaining cells are alive, which will be a really low rate, probably like much less than 1% of all cells. And you want to take those and inject those into a white mouse zygote. So it will bind to the dividing cells, and you know that it is from a brown mouse. So when this zygote develops into an adult, it will have these patches, these brown patches on it, and that means that it's a chimeric mouse. So it has two different genomes inside the same organism. So what you want to make sure that happens is that it got the brown mouse cells are in the gametes, so the sex cells. So what you're going to want to do is try and crossbreed this with a wild type or white mouse, and from the progeny you can tell whether or not it was correctly inserted. If it's a normal white mouse, then that means it might not have been inserted correctly, or if it's a brown mouse, it has been inserted, and this mouse will have the desired trait that you tried to insert. So that's it, and I might go into detail later.